Rare diseases are those that affect no more than 5 in 10,000 people. However, these diseases are not rare in their entirety. It is estimated that 4 million people in Germany are affected. A total of around 8,000 rare diseases are known to date, and more than 250 are discovered every year.
Rare diseases present themselves as, with only in a dozen or several hundred people worldwide, or patients worldwide. So the problem is that finding new drugs is very expensive and very time-consuming, and most people are not willing to invest that kind of money. So our approach to that is that we really we try to repurpose existing drugs. So we try to take a drug that is known and that is safe and try to use it on people that suffer from rare diseases.
The development of a drug usually takes 10 to 15 years and costs around 2 billion euros. Less than 5% of all drug candidates are actually beneficial for patients after clinical testing. That's why Ole Pless, a scientist at the Fraunhofer Institute for Translational Medicine and Pharmacology, uses a trick to find active substances against rare diseases.
So here we are focused on drug repurposing. That means that we take already approved and marketed drugs and we use them for different indications. And this trick allows us to speed up the development quite significantly and also reduce the costs. So we are focused on screening. That means that we kind of sieve through very large collections of chemicals, and most of these are not active. But we just look for some, like looking for a needle in a haystack, that actually show an activity and that are beneficial to treat a certain disease.
The search for the needle in a haystack is being carried out systematically at the Fraunhofer Institute in Hamburg with a library of over 5,000 active substances and with the help of robots and artificial intelligence, as well as human intelligence. The process is complex.
The new thing about these stem cell models is that they can really recapitulate certain disease aspects from the patient in a tissue culture dish. Of course, they do not represent the entire patient, but certain disease-relevant aspects that we can then try to make better with our drugs.
These models reflect the molecular basis of the disease and thus serve as model systems for finding active substances. It's a game of unknowns. Will one of the many thousands of active substances cause a reaction in the test? And will the reaction, the hit, be useful in terms of therapy? Thousands of active ingredients are placed on the transport containers with the stem cell models and are observed. As with a loading terminal, the systematics, logistics, and repeatability of the processes are particularly important to be able to assess the effectiveness of drug candidates. Finally, computer specialists are needed to analyze the data. The research group led by Ole Pless has made it possible to treat a rare disease in a completely new way.
Yeah, our team has identified a certain drug, which is called Viagra, which is well known, that this is beneficial for patients who are suffering from Lee syndrome, which is a metabolic disorder. So the powerhouses of the cells are affected, so the mitochondria. And using this Viagra drug, we could see that actually the patients benefited a lot from that in a clinical setting.
A huge advantage of the so-called repurposing, which means the repositioning of existing drugs, is the shortened approval process. Since Viagra has been on the market for many years, the risks and side effects are well known, also in children. The active substance can be administered directly to patients with rare diseases by experienced clinicians without having to undergo many years of testing beforehand. This can be life-saving for patients.
Our goal is to bring new medicines to patients and to find medicines to treat patients that are currently untreatable. And stem cell models can facilitate this process because we can maybe reach that quicker and we can deliver more safe drugs to these patients in the clinic.